My name is Sofia Mayans and I'm the CEO of InfiCure Bio. InfiCure Bio is a young startup located in the north of Sweden. And we have a focus on fibrosis, since fibrosis is a huge problem. It contributes to more than 40% of deaths in the industrial world. And despite this huge impact on human health, no efficient antifibrotic drugs are currently available on the market. Fibrosis is scar tissue formation within an organ, and this scar tissue is formed due to organ damage. And you can damage your organs in different ways. You can smoke, eat too much fat food, drink too much alcohol, or have a disease. Fibrosis can be formed in any organ, and if enough fibrosis is formed, the organ will completely stop working. We know that more than 90% of big pharma are developing drugs to treat fibrosis, but something is holding this development back. And one thing is that there is a need for good and reliable preclinical models in which these pharmaceutical companies can test the effect of their new drugs. Since when you develop a drug, you have to test the effect of your drug in an animal model before you can test it in clinical settings in humans. And this is exactly where we come in. Since we have developed this NIF mouse, it's a model that spontaneously develops inflammation and fibrosis of, for instance, the liver. This mouse was developed by me and my co-founder of the company, and we are the only ones in the entire world who have access to this mouse. So this mouse is the thing that we offer to our customers. So our business ID is a fee for service. So what we do is that we offer our clients, pharmaceutical companies, big pharma, academics, or basically whoever that is developing drugs to treat fibrosis, to test the effect of their new drugs on the fibrosis in our NIF mouse. So today our customers send the drugs to us, and we do all the treatments of the animals, we do all the analysis, and then we report back the results to our customers. In the future, when our patent is approved, we will start to sell licenses. And this means that the clients that are buying a license, they will actually get some mice that they can treat themselves and do all the work themselves. So this really makes our business idea very, very scalable. So our vision is to become a first-hand choice for tests of antifibrotic drugs. Of course, there are competitors out there. There are other animal models that can use, but common to these other models is that you have to actually harm the animal. You have to treat it with either chemicals or do surgical or surgery to induce this organ damage and then the development of fibrosis. So drawbacks with these methods, the competitive methods, is that you have a weak resemblance of fibrosis. You have a weak resemblance to the human condition. You have problems with reproducibility, and it's very both time-consuming and hard to actually induce this fibrosis in a good way. So advantages with our NIF mouse is that, as I said, we have a spontaneous development of fibrosis. This occurs at an early age, and this means that we can actually shorten the time, the test protocols for our clients. We can save time for clients, and this is very important if they have a patent for the drug they are working on, because then they want to save patent time. We do also see that the development of fibrosis that we have in our NIF mouse is fairly similar to what you see in the human situation, since we have a chronic inflammation that is preceding this fibrosis, and we have 100% reproducibility. And this means that when we look at the livers of NIF mice at the same age, we can see that we have the same levels of fibrosis. So when we treat one group of mice with a drug and one group of mouse with just a control, and we see a reduction of fibrosis in, for instance, the group where we're treated with the drug, we know that the reduction in fibrosis is actually due to the drug and not due to variances in levels of fibrosis to start with. So we have already introduced this NIF mouse as a liver fibrosis model. We have signed 9 customer contracts and 1 collaboration contract. And we estimate that our sales will take sort of really progress during 2019. We will get 50 new contracts. In 2020, we'll start to sell licenses because we estimate that our patent will be approved in the end of 2019. And by 2023, we will have a revenue around 150 million Swedish crowns. So, the funding so far, well, in the beginning we had a beneficial loan from the biotech incubator here in Umeå in Sweden. We have also revenue from customers, of course, from our customer contracts. Local investors invested in the company in end of 2017. And we have also obtained grants from EU and from Vinnova. Thank you very much for your time.